everyone. Today we will discuss about the hybridoma technology. So let's begin with the class. So as we know that hybridomas are the it is a kind of hybrid of two cells. Okay, so hybridomas are the cells that have been engineered to produce the desired antibody in a large amount. Okay, so when you fuse one cell to another cell, it will make a hybrid cell which is called as the hybridoma. Okay, here it is called as the hybridoma. This cell is called as the hybridoma. Okay, and why we need this hybridoma cell? Because we wanted to produce certain kind of desired antibody, desired product in a large amount. Okay, so that is the hybridoma technology. Okay, so hybridoma technology is a method for producing large number of identical antibodies, also called as the monoclonal antibodies, which is specific to the antigen of interest. Okay, or we in another word we can say like hybridoma technology is a method for producing large number of identical antibodies, and this process is start by injecting a mouse with an antigen that provoke an immune response. Okay, and this process how we do generally do is by injecting the antigen into the mouse model. Okay, to produce the larger amount of antibody against that particular antigen. Okay, so that is called as the hybridoma technology. So hybridoma cell line are formed via the fusion between a short-lived antibody producing B cell and an immortal myeloma cell, and each hybridoma constitutively express a large amount of the specific monoclonal antibody. Okay, so the principle behind of this hybridoma cell technology is that it is having two different cell. Okay, two different cell. One cell is having the antibody producing capacity. Okay, here suppose it is having the antibody producing capacity, and this cell is having the immortality. Okay, when you fuse these two cell together, it the hybrid cell will consist both the nature. Okay, means it consists the nature of and continuous antibody production as well as the immortality. Okay, so this is the basic principle behind the hybridoma cell technology. Okay, and generally we use this hybridoma cell technology for the desired product production in a larger amount. Okay, or we can cryopreserve this product for the longer time. Okay, so as like this hybridoma cell technology is generally used for the monoclonal antibody production. Okay, so what is the difference between monoclonal and polyclonal? Okay, so yeah, so first thing is monoclonal antibodies, it is as the name suggests, mono means single. Okay, here you can see that this is the structure of the antibody. Okay, this is the monomeric structure of the protein. Okay, so and this CDR region, or we can say like antigen binding site, is having the means I can say like same kind of antigen determining site, these two. Okay, and what is the polyclonal antibodies? Suppose in your blood there is different type of antibodies present. Okay, and each antibody is having specificity toward different type of antigens. Okay, here the monoclonal means this antibody is having single specificity toward the antigen. Okay, suppose this is the very big antigen. Okay, and this sorry, this part is having one epitope, this part is having one epitope, this part is having another epitope. Okay, so what is the monoclonal? Monoclonal will detect only this antibody. Okay, monoclonal will detect this antibody, this antigen. Okay, this part of the antigen it will detect, but polyclonal what it will do is it will detect this epitope as well as this two epitope also. Okay, so the monoclonal antibody, it is the single specific population, I can say. Suppose this is the serum. If you are talking about the monoclonal antibody, means whatever antibody present here, it will specifically recognize this particular epitope. But if you are talking about the polyclonal antibody, then whatever population present in the serum, that can recognize variety of the epitope in a antigen. Okay, so that is the basic difference. And this monoclonal antibody are the antibodies that are identical because they are produced by only one type of cell, okay, immune cell, and all clones of a single parent. Okay, and basically produced by the white blood cell, that is called as the plasma cell. Okay, so plasma cell is basically produce the not monoclonal antibody, it produces the antibody. Okay, and it is used for the treatment of cancerous cell and the antivenom. Okay, so the difference between the monoclonal and polyclonal is, it is like it recognizes a single epitope of an antigen, and it recognizes the multiple epitope on an antigen. Okay, and the production is continuous and uniform. Okay, because it is not having any biasness or any randomness. It means specifically determine only single epitope, so that's why it is continuous and uniform. Whereas this different, in case of the polyclonal antibody, different batches vary in composition. Okay, as I earlier told you, that if suppose this serum is containing the polyclonal population of antibody, so this all antibody can recognize different type of antigens, right? It will be the cocktail of the antibodies. Okay, but in case of the monoclonal antibody, it is all the antibodies present in a single population. It will recognize only a particular type of epitope. Okay, and it is expensive production because this we generally do in the artificial condition. Okay, and it require longer time for the production. Okay, but from here you can produce the large quantity and specific antibody. Okay, it is the inexpensive production because naturally the polyclonal antibodies is present, and it is the rapid production because in our body it is producing, so we no need to like do this process in the artificial condition, right? And from here also large quantity can be produced, but for the longer storage we can't prefer this polyclonal antibody, as well as it is non-specific antibody. Okay, so this is the difference. So as I told you, what is the principle of hybridoma technology is, here one cell which is called as the plasma B cell, okay, which is producing the antibody, and one myeloma cell which is cancerous cell, I can say. Okay, this is the cancerous cell, and this cancerous cell is immortal in nature. Okay, and when we fuse these two cells together, it will create the hybridoma cell or hybrid cell, which is having the property of antibodies production as well as the immortality. Okay, so the hybrid cell has the capacity of antibody production derived from the B cell. At the same time, it can divide continuously by the quality derived from the myeloma cell. Okay, it can divide for the infinite time because it is immortal in nature, and this characteristic comes from the myeloma cell. Okay, by combining the desired quality of both the cell, the technology ensures this high antibody production of single specificity. Okay, and this specific hybridoma yielded monoclonal antibody in artificial media, and this whole technique is called as the hybridoma technology. Okay, so the basic principle behind this is the plasma cell and myeloma cell fuse together and produce one hybridoma cell which is having both the properties. Okay, so this is the hybridoma technology. So now how we perform the hybridoma technology, this is the complete procedure. Okay, so for that what we have to do is, firstly we have to immunize the animal. Okay, immunize the animal by against which you wanted to raise the antibody. Okay, so that antigen you have to inject in the mouse model. Okay, and then what will happen in spleen, it activate the B cell to produce the antibody against those particular antigen. Okay, that convert into the plasma cell and memory cell. Two type of cell it will form. It will form plasma B cell and memory B cell. Okay, so this plasma B cell will produce the antibody against those particular antigen. Okay, and then you have to isolate this plasma cell from the spleen of the animal. Okay, and the second step is isolation of the myeloma cell. Okay, firstly you have to isolate the plasma cell, and then you have to isolate the myeloma cell. Myeloma cell are the cancerous cell which are isolated from the bone marrow. Okay, and myeloma cell are generally immortal in nature and has the multiplication capacity. Okay, so that we can isolate from the bone marrow of cancer patient we can say, or cancerous cell we can say. Okay, and this is having immortal in nature. Okay, then the third thing is fusion of the spleen cell and myeloma cell. Okay, so through the fusing agent, that is the polyethylene glycol. Okay, we fuse these two cells together. So there is here you can see that plasma cell and myeloma cell, when we fuse together with the help of polyethylene glycol, there are five probability to produce cell. Okay, the first probability will be fused plasma cell. Okay, so plasma cell will can fuse together and form the fused plasma. Okay, likewise the myeloma cell can fuse together and form the fused myeloma. The third one is the combination of these two, plasma and myeloma, that can create the hybridoma cell. The fourth one is the some plasma cell can't fuse. Okay, so will not fuse, so it will be remain as a unfused plasma. Okay, likewise some myeloma also will not fuse, so that is remain as a unfused myeloma. Okay, so there are five probability: two fused, two unfused, and one is the hybrid cell. So this hybrid cell you have to isolate in the next step. Okay, because these two you don't need. Okay, and only this cell is having the property of antibody production as well as the long term multiplication. Okay, so how you will isolate from all the five probabilities? In fourth step, we are having the selection of het media. Okay, so there is the het medium. Okay, which is called as the hypoxanthine aminopterin thymidine. Okay, so what is the quality of this het media is, this aminopterin is work as a inhibitor. Okay, so this aminopterin is a inhibitor for the de novo synthesis of nucleic acid synthesis. Okay, so like here it is it has been told that before multiplication of cell, it is necessary to synthesize new copy of DNA. For that they require the synthesis of nucleotide. Okay, so nucleotide synthesis can be done in by the salvage pathway as well as the de novo pathway. Okay, so in case of salvage pathway, require degraded part of the old nucleotide to produce the new nucleotide. Suppose there in the media, if there is a having degraded nucleotide, then from that degraded nucleotide it can produce the new DNA or new nucleotides. Okay, and in case of the de novo synthesis, it is completely new nucleotide molecule. Okay, synthesized in case of the de novo. Okay, so when your de novo is not working properly, that time salvage pathway play a role to producing the nucleotide. Okay, so in this het medium, what is happening is, so in the het medium, cells are not synthesized by the de novo synthesis. Okay, because of the presence of aminopterin in the het media. Okay, so this aminopterin is the work as a inhibitor for the de novo synthesis. Okay, and which block the dihydrofolate reductase enzyme, which is necessary for the nucleotide synthesis. Okay, this is the DHFR enzyme. Okay, so this is the DHFR enzyme. Okay, and this DHFR enzyme is inhibited by the aminopterin. Okay, so cells is having two pathway. Okay, first is having the de novo and second is the salvage. Okay, so de novo pathway is blocked because of the presence of aminopterin. Okay, so there is only one pathway left, that is the salvage pathway. Okay, so in the for the synthesis in salvage pathway, cell require the HGPRT enzyme. Okay, so this salvage pathway is required the HGPRT enzyme. Okay, so what we generally seen is like here. Okay, so what we seen is HGPRT, and this immortal cancerous cell is we engineered in such a way that this cell, it is not having the HGPRT enzyme. Okay, it is not having the HGPRT enzyme, whereas this antibody producing B cell is positive for the HGPRT cell. Okay, so what will happen, already the de novo synthesis is blocked because of the presence of aminopterin. Okay, now because of the absence of this HGPRT, the myeloma cell will not survive in the het media, because it can't also not perform the salvage pathway, because this HGPRT is responsible for the salvage synthesis, right? So if both the pathway is blocked, then this cancerous cell will not survive in the het media. Okay, whereas this mortal B cell is having the HGPRT enzyme, so that's why it can survive in the het media. De novo synthesis will be blocked, but this their salvage pathway will work and provide it the nucleotide. Okay, but in case of this normal B cell, what is happening is it is having certain half life. Okay, so after certain time it will die. Okay, so this both cell will automatically die. This is because of the absence of HGPRT, and this is because of the short half life. Okay, whereas this hybrid cell, which is having the property of both the cells, and it is HGPRT positive as well as HGPRT negative, it will survive because it is having the capacity of both the cells, and it can produce the antibody, as well as it can multiply. Okay, so this is the whole idea of the het selection. Okay, and likewise there is another enzyme, that is the thymidine kinase, but most major enzyme is the HGPRT. Okay, so this thymidine kinase is also same, like it is this HGPRT will work on the hypoxanthine. Okay, so here in the media for the nucleotide synthesis, two component is there. One is the hypoxanthine and second is the thymidine, and this aminopterin is the inhibitor of the de novo synthesis, and these two will play for the nucleotide synthesis as a precursor for the salvage pathway. Okay, so HGPRT will work on this hypoxanthine, and thymidine kinase will work on this thymidine. Okay, so like that, this thymidine kinase is also negative in case of the immortal cancerous cell, and it is positive in case of the normal B cell. Okay, and these two properties is having in case of the fused hybrid or hybridoma cell, that's why it will survive and it can multiply into the different hybridoma cells, and it can produce larger amount of the antibodies. Okay, so like that we have to like screen. Okay, so once it this selection process has done, now we have to screen the hybridoma cell for desired antibody production. Okay, so ELISA screening method is done by incubating hybridoma culture, in which secondary enzyme gets conjugated and formation of the colored product shows the positive hybridoma. Okay, so suppose these two cells is fused, but it is not producing your desired antibody, so that selection also we have to do. Okay, so for that ELISA we have to perform, and if it is giving signal, so that's why that means it is producing our desired antibody. Okay, so this is the complete procedure of the hybridoma technology. Firstly, we have to immunize the mouse with antigen against which we wanted to raise the monoclonal antibody. Okay, then we have to isolate the myeloma cell. Okay, and after isolation of the myeloma cell, we have to fuse through the PAG and DMSO. Okay, this two cell fused and DMSO, then there is five different probability of cells. Okay, unfused myeloma as well as unfused B cell, fused and fused myeloma, fused B cell, right? So this fused cell and unfused cell will be die because of the these two, and these two cell will die because of the like this is because of the shorter lifespan, and this is because it is not having HGPRT enzyme in their nucleus, right? So that's why it can't survive in the het media, whereas this fused myeloma will survive because of the presence of HGPRT. Okay, then we will isolate this screen of positive screening clone by ELISA. Okay, then isotyping of the generated antibodies, and for the larger quantity production of antibodies, we can perform the in vitro, and then we can do the in vivo. Okay, so in vitro propagation will give you the low concentration volume, that is 1 to 20 microgram per ml. Here it can give you the high concentration, that is the 1 to 10 mg per ml. Okay, then here you have to do the purification of monoclonal antibody. Okay, in case if you are doing from the in vivo, then you have to do the purification of monoclonal antibody. Okay, because in this mice, also in the body, there are polyclonal antibodies. So for isolation of monoclonal antibody, you have to tag your antibody with a certain kind of tag. Okay, like his tag and GST tag. Then you have to like protein purification, you have to purify your protein antibody through the column, and then characterize it and store it. So, in the question, it has been asked like production of monoclonal antibodies by hybridoma technology require. Okay, so option is splenocyte, osteocyte, hepatocyte, and thymocyte. Well, what will be the correct answer? So, the correct answer of this question will be splenocyte, right? Splenocyte is the cells present inside the spleen. Okay, and osteocyte is present in the bone. Okay, and this hepatocyte is present in the liver, and this thymocyte is present in the thymus. Okay, so we generally isolated the B cell from the splenocyte. Okay, so the correct answer of this question will be splenocyte. Okay, so thank you all for your time.